Hello everyone, I made this video so that I could explain how we can assign gene functions while we're doing our annotations. So when we are given our gene sequence, we are given a sequence of DNA and we choose the correct reading frame, and each of these three codons, when the DNA is, when transcription occurs to synthesize the messenger RNA using the DNA as a template, we have codons that correspond to amino acids, and these amino acids are linked together by peptide bonds to form the protein. So this link from DNA to RNA to protein is called the central dogma. Today we're talking about proteins, so I wanted to make sure that we understood something about structure of proteins so that we can then correlate structure with function. So we have the primary sequence. So if I go back to this diagram right here, the sequence of the bases on the DNA determine the sequence of amino acids in the protein. So we have the primary structure, which is the order of the amino acids in the protein. And there are elements of the primary sequence that can fold up to form a secondary structure, which is a regular repeating type of structure. The most common examples are the alpha helix and the beta sheet. The alpha helix is shown here. The entire protein chain could fold, with the secondary structural elements being part of this, can fold into a tertiary structure or a three-dimensional structure that has a specific shape. There are some proteins that associate with multiple protein chains to form quaternary structure, and we can see this in our phage proteins. There are times when multiple copies of a protein will be made, and then they will associate to make higher order protein structure. So structure is linked to function, so that is why we're interested in this. So just to define some more terms, protein domains. This is pyruvate kinase. It is a single polypeptide chain, but the artist has colored this in three different domains. You can see that each of these could fold on its own and is a distinct entity kind of linked with these loops right here. Many proteins consist of several domains like this one, and you can imagine domains as being something like squares of patchwork quilt, and you can take those squares and you can stitch them together, and you can use different squares and stitch them together differently to make proteins, and these proteins would have different structures. But maybe if you picked the same domain, then you could make a family of proteins with similar functions. So this isn't on the C phages list, but we can use JPRED to determine if we have helix-turn-helix domain proteins. So the helix-turn-helix is important because this picture shows the DNA over here with the backbone in orange, and this shows a protein dimer, and one dimer is shown in blue and one is shown in pink, and they showed the blue subunit of the dimer in a darker blue to indicate the portion of the protein that binds to the DNA. So the helix is shown as a corkscrew, so I don't know if you can see this helix-turn-helix right here in dark blue. The other member of the dimer is shown in pink, but they colored it in red to show you the portion that binds to the DNA. I think this is more clear. You can see the helix and then a turn and a helix. So the helix-turn-helix is the portion that binds to the DNA. Here you can see that the helix-turn-helix binds to the major groove of the DNA, but there are also other domains of the proteins that are linked to function. So DNA has a fairly regular structure, so you can imagine that a helix-turn-helix could bind to the DNA, but then these other domains right here would specify the function. We do have a protein called JPRED, a program called JPRED that can help us, let me go back here, determine if we have a helix-turn-helix DNA binding domain. Oh, that's for membrane protease. Let me go back here. So I think I have a JPRED open window here that I ran before I did this. So this is for Frankenweenie. This is gene product 230, and we can see from the blast that we get some hits to helix-turn-helix proteins. But just to be sure, we can take the product and we can copy it and then put it into JPRED, and what you do is you just copy over what's here and then paste I like using the advanced options because you could put in your email address, and actually you can see I've already run G protein 230 before. So I'm going to go ahead and hit make a prediction. So what happens is sometimes JPred can take a while, and then you can go on and do something else, and it will just send you an email, send you a link to your email. So here is the data for JPred for G product 230. So we found that if we view simple results in HTML, that was really helpful. So what this does is it lines up the amino acid sequence on the top using the one-letter codes, and you can see that we do have these helix, turn, helix domains. So H means helix, and the turn is usually about 4 amino acids. So you can see where these dashes are. So we have a number of helix, turn, helix domains in this protein. So we can confidently call this a helix, turn, helix binding domain. All right, so let me go back to my PowerPoint then and pick up where I left off. All right, so we've done JPred. So the next thing that we want to do is we want to look at if we have possible membrane proteins. So this is G protein 113 in Frankenweenie. So we have 3 different programs that we use to determine if this is a membrane protein or not. So let me go ahead and exit out of here. And okay, we can close JPred now. So here's SOSUI. So see, I've already copied this in, so I can hit execute, and this is usually pretty fast. So the way that this works is that they look at the amino acids, and amino acids are given different scores depending on how hydrophobic they are. And you might remember that the inside of a membrane is very hydrophobic. So here, SOSUI has predicted that there are 4 different transmembrane domains. So maybe this protein sort of snakes across the membrane and goes back and forth, back and forth, back and forth, and that 4 times it crosses the membrane. So SOSUI says that this protein is in fact a membrane protein. We can do the same thing in TMMHM, and you see I've already pasted this in. They need this to be in FASTA format, so you'd have to use this greater than symbol and the 113. So let's go ahead and submit that, and again, usually that's pretty quick too. Now here, the number of predicted transmembrane helices is 3. That's because TMHMM has sort of lumped these together, maybe figuring that there's not enough space for this domain to go back and forth across the membrane. But we can still conclude that this is a membrane protein. We can do the same thing for TopCons. This also allows you to email the results to you if you like, if you think that it's taking too long. But actually today things are pretty quick. So TopCons gives you this nice graphic where it shows inside, outside. The transmembrane helix from in to out is in gray, and from white it's out to in. So here we're going, the protein is inside, goes across to the outside, and comes back. And here, again, maybe there are 4 membrane domains, maybe there are only 3. This is a nice visual where you can see like maybe there's not enough space. So so this is maybe a little bit difficult to tell. But we can see that this protein is a membrane protein. So one of the things that we should notice when we look at our genome is that we have sort of groups of genes in different places in the genome. So on the 5' end, you usually have the virion, structural, and assembly genes. So these are the genes encoding proteins that are part of the phage itself or assisted its formation. So terminase, portal, capsid maturation protease, scaffolding proteins, major capsid protein, major tail subunit, tail assembly chaperones, tape measure protein, and minor tail proteins. If we continue along the genome, let let me show this picture first. So, so here's a phage particle right here, and this shows the capsid. So again, we'd make multiple copies of this protein to form the capsid or the head, where the DNA is located. The portal protein and the head-to-tail connector proteins will connect the head to the tail. The tape measure protein determines the length of the tail, and there are proteins here that form the major tail proteins. At the tail tip, we have minor tail proteins, and we might also have some hits to some tail fibers, which are minor tail proteins. So then we see that, again, grouped into another area of the genome, we'd have genes involved in phage DNA replication, because the phage needs to make copies of its DNA. So DNA polymerase, DNA primase, DNA helicase, nucleotide metabolism genes, and single-stranded binding proteins will all be here. There are also genes involved in life cycle regulation, so repressors, activators, integrases, and repressors. We would see those too if the phage is able to adopt a temperate lifestyle by forming a lysogen, recombination directionality factors, things like that. And finally, genes involved in lysis. The phage have to get out of the bacteria once it has made copies of itself, and to do that it needs to lyse the bacteria. So there are proteins involved in lysing the bacteria so that the phages can escape and go on to for another round of infection. There are other well-characterized genes, transcription factors, toxin, antitoxin systems, peptidases, phosphatases, host gene homologs, methylases, nucleases, and DNA binding proteins. We don't always know what the role of these proteins is in the phage, even though we can identify them. So what we should do is we should go to the bioinformatics link. Let's just skip verification. Go right to the bioinformatics guide, and here we should read this article. So these are functions present in almost all genomes. So there's a nice article right here where there's a ranking of function. 1 must be present in the annotation, 2 is found in most but all, and 3 maybe you can find it and maybe you can't. So you see for terminase, all tailored phages will have terminase. So terminase is the motor that packages the DNA. So every genome should have 1, but not more than 2. Maybe there's a large and a small subunit. So that's one example. The capsid maturation proteins, maybe you'll be able to locate it and maybe you can't. This is a nice article that we should read to inform our functional assignments. But even with all of the things that I've showed you, we can only assign functions to somewhere between 10 and 30% of the genes, and that's totally normal. So what are the other tools we can use to make functional assignments? Well, one is we can blast. So we can blast within DNA master, and we can also blast in phages DB in the gene list on the phage page. So if we do this, actually let me do this first. So let's exit out of here. So, for example, for each feature in DNA master, there's a blast that's done within DNA master. But you can also go to the database. So, for example, this is for Frankenweenie, and this is gene protein 9, and you can go to NCBI and there will be a link to it. But you can just, if you want to, you can blast it right on phages DB. And that will give you just phage proteins. And the other thing it will give you is it will give you anything that's a draft. So Frankenweenie's best hit is to itself, but we also have some nice coverage of the other phages in this genome. Now, it does suddenly occur to me when I look at this that we might need to go back and look at the start for Frankenweenie, because we have an extension here that we don't see in some of the other phages. So maybe that's the correct start, but I think we should go back and look at that again. So we can blast within DNA master, we can blast in the gene list on the phage page like I just showed you. We can also look at we can actually go right to the NCBI blast server, and that's nice because you can see some visuals that you don't see on DNA master. And we should also keep in mind what we're doing this. So there are ways of doing the BLAST P searches. So again, if you do this within DNA Master, that's the non-redundant database. It includes phage proteins and non-phage proteins. If you go to the NCBI BLAST server, you can use a experimental database, so you can actually use a different database, or you can blast, just like I just showed you, within the actinobacteriophage database on PhagesDB, because that will give you only phage proteins, and it will not give you anything that's not a phage protein, and it also will include draft sequences. If you go to the NCI BLAST server, depending on which button you check, you could do the non-redundant database and the conserved database. So this is GP9 and Frankenweenie. So if your BLAST P gives you a significant result against the conserved domain database, you'll get something up here. So this is just a screenshot. So let me go back and do this live. So I've just done the BLAST search on the NCBI website. So I did this on, I did a BLAST P, and these are the results that I got. And you can see we get the hits to other phages that are in GenBank. I like the graphic summary because it shows the coverage, which is good. But the other thing is that since we're using the conserved data domain database, if you go up here, you can actually, if I go back to description, this is going to help us with our functional assignments, this Thi-X-like thymidylate synthase, and other hits to an FAD-dependent thymidylate synthase. So if we go back to the graphic summary and we go to the conserved data domain database, then this will give us a really nice graphic of, again, very nice coverage. And if you sort of mouse over, so thymidylate synthase, so lots of hits to thymidylate synthases. So these are FAD dependent. That's why we have some hits there. This can also, looking at these the conserved domain database in when you do a protein, a BLAST P, this can be a really nice visual to help us with functional assignments, and you don't get that if you're on DNA Master. So the experimental database, so if you can do the standard non-redundant database, that takes a little longer. I'm liking the experimental database because this is a link to a curated website, UniProt. So that means if there's multiple copies of a submission, that happens in GenBank, where you have lots of different copies in GenBank, but the curated website will make sure that there's only one. So there's that. But the other thing is that it puts 90% related proteins into clusters and then chooses a representative protein from that cluster. So, and it's long enough so you won't miss anything, but you just blast against that representative, so the blast search is much quicker. So I've been using the experimental database recently. So if we're going to assign functions, we should look at the e-value. So it should be 10 to the minus 7th or less for functional matches. It can be less stringent, 10 to the minus 4, for other purposes like calling start sites. The closer you are to 0, that means the less probability that this is just by chance. You want to look at the length of the alignment. Does it extend the entire length? If it doesn't, maybe you just have a domain. Maybe you don't have the other function. Maybe you don't have the active site if it's an enzyme, but you can use that to refine your start choice. And you saw that with G protein 9, where we should go back and look at that and make sure that we've called that properly. And we should look to see if the match is either phage or bacterial in origin. If we get something to animals or plants or humans, that's not likely to be real. So the other thing that we can do is we can set up along with our BLAST, is we can set up a Famirator window, and many of these published genomes will have functions labeled. So, for example, here we are with our Frankenweenie draft. So none of the functions are called here because it's our job to do that. But if we look at just because, we can see that this gene is in the same fam. It's 6, if I can read this properly, 6568. And you can see in just because and in Keela and Satis and Cradle and I don't know how to pronounce this, EI Ili Mejor, it's all classified as the terminase. So that can help us with our functions as well. The other thing we can do is that there are also links to the conserved domain database here. So if we click on terminase and we click on domains, now you don't always get something. This is just a little hit right here. Okay, so this is part of an endonuclease. So this is actually not particularly helpful. There are other occasions where looking at the domains will be much more helpful. Here's an example where domains are more helpful. So here, this is protein is called as a glycosyltransferase. So if we click on that, then so here, each one of these orange bars is a hit to some kind of domain. So here we have a glycosyltransferase domain here, but there are quite a few domains here. Again, if you click on it, it will give you the alignment and show if it aligns to the entire protein. So again, this particular gene has quite a bit of information. So what you do is you just click on one of the domains, and then it will give you information about it. So again, using the domain function in Famirator, you could get hits to the conserved domain database, and that can be helpful. Here's an example with another phage we were working with earlier. We called this function correctly as a DNA methyltransferase, but if we look at these domains, we found that we have some additional information. It's an adenine-specific DNA methyltransferase, so it can give you additional information. So BLAST can help us, Famirator can help us, and of course there's HHPRED, where we're looking for conserved domains. So, for example, this is for Frankenweenie, this is GP, gene protein 9, and we get some nice hits again to the thymidylate synthase. So we get some nice coverage, and the other thing is that we also get hits to here, let me go up here. If you see a 4-letter alphanumeric code, sometimes you'll see something like this, a C, like it's a C subunit. This is to the protein data bank. So you can click on this. So this is Mycobacterium tuberculosis. That's interesting. So this is to the thymidylate synthase. So again, this can help our choice because this is an actual crystallized protein. The other thing is if you go over here, it shows you the biological assembly, and it shows you the asymmetric unit, which is just one of the proteins, and then if it assembles to form a multimer, it will show you that as well. So here we have a probability that's 100%, which is pretty good. We have lots and lots of hits to lots of different thymidylate synthases, and not all of them are on the protein data bank. Some of them are on the PFAM site, but a lot of these are to the protein data bank. We have E values that are very, very low, and we have coverage that's also very good. Now that's not always going to happen. So for example, if we look at G protein 10, for example, oh, that wasn't that I wanted to show. Okay, that is actually here. This is what I wanted to show you as an example. So here we don't get a lot of hits. We get to hits to DUF is domain of unknown function. So if we go down here, we do get some hits above 90%, but if there are families of unknown function, or things that might be relative to phages like collagenases. this is probably not going to give us enough information for us to call the function for this particular protein. And the C phages people really don't want us to use anything at all that's less than 90%. So that would be these top 4 hits right here. And also, if you see the E values, those are very, very high, especially if you compare them to G protein 9. So we probably wouldn't be able to use HH-pred to call the function in this case. So when using HH-pred, like BLAST, some matches are very useful, others are false positives. So good matches have probabilities of 80% or greater, and they will be red, and the bluer they get, the less good the matches are. The scientists who wrote HH-pred claim that matches over 30% might be real, but our friends at Pitt say you can't reform functions unless you have a probability over 90%. So we will not do that. I've also noticed that if you look at the databases that HH-pred uses, we have the conserved domain database, we have the UniProt, that's the curated database. We have the protein data bank, which is crystallized proteins that people have done experimentally in the lab. And then we have PFAM, which is more bioinformatics, where you're looking at sequence to see if it lines up with anything proteins that are already known. So HH-pred doesn't, though, rely solely on amino acid sequence, but it tries to capture the patterns generated by the 3-dimensional shapes. So you need to look for 90% or better, good, really low E values, and the function stretches the full length of both proteins, both your protein and the protein that it's aligning with. So the last thing is synteny. So synteny, a lot of the genes in bacteriophage genomes appear in the same order. So for example, if you look at the 5' end for the proteins that code for the proteins making up the virus particle itself. So we have terminase, portal, capsid maturation protease, scaffolding, minor capsid subunit, major tail subunit, tail assembly chaperones, tape measure, and minor tail proteins. So there, with phages, you never know. There can always be some variation, but we shouldn't have anything that's wildly different from this. So this is from other information on the bioinformatics website that I showed you earlier. So again, we should look for terminase, portal, capsid maturation proteins. There's a lot more detail on the website that tells you, is there one? Is there more than one? Should we be able to identify it? Do all phages have it? Should we make sure we have it in our genome? So here's an additional major capsid subunit. Tape measure is always the longest gene in the genome. There's only going to be one. The tail assembly chaperones are upstream of the tape measure. Sometimes there's a translational frameship, not always. Minor tail proteins are downstream. So knowing the synteny of the order can help us with our functional assignments. So before bioinformatics, the only way to determine gene function was to perform wet bench experiments, cloning an expressing gene or knocking a gene out and then characterizing the resulting mutants. These kinds of studies are still the gold standard for determining gene function. So here is an experiment that was done in the Hatfield lab where bacteriophage Marvin was isolated, and then they isolated the proteins and ran them down a gel, and here are molecular weight markers. And you can see that that's really difficult. You have lots of proteins right here, but you can only identify a small subunit of them, and here again you're identifying them by the size of the protein that's expressed. So as more recent genomes have better functional assignments than the older ones, and remember, our gene calls are based on the calls that scientists have made in the past. And finally, you have to use the C functions official list. So this was updated today. So whenever you make a functional assignment, you want to be sure that it is on the list. And one final thought, we can look and see what others have done, but sometimes they can lead you astray. So this is when we are looking at Langerak, and if we look at G protein 46, this is a Cas4 family exonuclease. We incorrectly called it a RecB-like exonuclease helicase because if you see, everybody else did. If you look here at the gene in this fam, everybody else calls it that way. But If we looked a little more closely, we would have seen, if we looked at the domains, that the domains that are stringing across here belong to nuclease superfamily. This is another domain of unknown function, but it's associated with nucleases. And finally, here's another domain right here that is associated with a nuclease. So we should have called this an exonuclease. There isn't any helicase domain there. So these calls are incorrect. So sometimes that will happen, and that's something that we should keep in mind. So I hope this is helpful, and I hope this helps us do the best possible job we can to call the functions in our genomes.